Right. Hello, everyone, and welcome to today's CryoEM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zimani. I'm a scientist and training liaison at the National Center for CryoEM Access and Training, NCCAT. I'm joined today by staff members from each of the centers, including my colleague here at NCCAT, Shabangi Agarwal. Joining from the Stanford SLAC CryoEM Center is Nathan Burrows, and we're also expecting Michael Schmidt today. And here from the Pacific Northwest CryoEM Center, PNCC, is Kirsten Wheeler. And before we give the floor to our speaker today, we'll have a quick update from each of the centers on the resources we make available at no cost to the research community. S2C2 has invited today's speaker, Greg Pintilie, who's joining us from Stanford University, and Nathan will introduce him properly after our center updates. This webinar is a regular event we host the last Thursday of each month, and we highlight users of the National CryoEM Centers who come and generously share the tips and tricks that have enabled them to make use of CryoEM in their research. So you can save the date for our talk on June 26th. Professor Rebecca Page will join us from University of Connecticut Health to tell us about decoding mechanisms that control protein phosphatase specificity. And a registration link for that talk is already on the events page at cryoemcenters.org, where you'll also find recordings of all of our previous talks. Along with this regular online forum, there are a couple of upcoming opportunities to interact with staff from the centers at some upcoming meetings. So this includes sessions at both the ACA meeting in outside of Chicago in the middle of July and the microscopy and microanalysis meeting in Salt Lake City at the end of July. There are a couple of sessions I highlighted here organized by staff from the centers. And do note early registration deadline for ACA is coming up in a couple of days. So if you're planning to go there or didn't realize that's a great venue to meet other people in the cryo-EM community, think about that sooner rather than later. All right, back to logistics today. If there are any questions about accessing National Center resources, you can reach out directly to any of the panelists here using the chat. For questions for our speaker during the talk, please use the Q&A box. You can send your questions at any time during the talk, and we'll hold them for moderated Q&A at the end of the talk. So let's go ahead and get updates from the centers. Kirsten, I'll let you let us know what's new at PNCC.
Thanks. My name is Kirsten Wheeler. I'm the administrative coordinator for Pacific Northwest CryoEM Center. We also call that PNCC. We have two proposal types, one of which is our screening access proposal, which allows up to 2 users to come on site for up to 5 days of sample preparation and screening with staff. Then we also have our general access, which is 480 hours of microscope time that goes over 2 years. The time is utilized for screening and data collection. Our screening access is actually aimed specifically at low resource institutions, however. Approved general access proposals are also eligible for training workshops, which cover topics from sample preparation through to data processing. We're excited about 2 recently added EP workshops, one of which is for screening, and the next one for that is coming up in August. That'll be our first flagship version, and then that will be followed by our data collection workshop for EPU. And that will be in October. PNCC has 6 microscopes. We have an Arctica and with a K3 camera for screening and 4 Krios instruments for data collection equipped with K3 and Falcon 4i cameras. 3 of our Krios are equipped with Gatan energy filters, while the other 2 have the Thermo Fisher energy filters. Our Krios 1 is now updated with a Selectress X. The newest member of our microscope family, however, is the Glacios 2. We've been recently utilizing that for many of our users' screening. sessions, and the hope is for those to be screening followed by small data collections. For our sample preparation, we've got two Vitrobots and a Leica GP2. If interested in working with PNCC, please go to pncc.labworks.org and apply today. Our deadlines for proposals are the first of each month.
Great.
That's it for
Thank,
me.
thank you, Kirsten.
Yeah, absolutely.
Shibangi, let us know what's new at NCCAT.
Hi, so I'm Shubhangi, a scientist operator helping users to collect their data at NCCAT, which is the National Center for CryoEM Access and Training. We are located in New York, Manhattan. And like other national centers, we are screening, collecting, and training users uh, for data collection. We also have, we are on the instrument side, we are upgrading some of our instruments with newest energy filters and direct electron cameras or Selectress Falcon 4i. We are also having some changes in our workflow where if you are an active user or an upcoming user, and if you have not received the coded grid boxes, please contact us. Also, we are scheduling QC sessions before the data collection so that the operator and the user know exactly what they want, and it increases the access of data collection. And we also have a integrative structural biology meeting being held at ASRC CONY on October 9th and 10th, and the registration is open. It is available at Eventbrite. So please, and if you want more information, please contact us at nccat.nysbc.org. Thank you.
Thank you, Shibangi. And Nathan, let us know what's new at S2C2.
Hi, my name is Nathan Burrows. I'm the program manager for our exploratory specimen screening and optimization program. And we have the typical suite as the other two national centers of equipment and available resources and programming activities. The latest thing that I want to share with everyone is that we are planning a SPA model validation workshop at the end of October, from October 28th to October 30th. So if you want to save the dates for that, more details will be coming forth with soon. And that's the theme of our topic today, is model validation. So I'd like to introduce our speaker to everybody. Our speaker today is Greg Pintilie. He is a wonderful resource here at our facility and our university here next to Stanford. And I'm very looking forward to his talk today, and hope you all as well.
Okay, so can everybody see my screen okay?
Looks good, yeah.
Okay, great. So thank you for the introduction, Nathan. I'm Greg Pintilie. I'm a research scientist in WACHU's group at Stanford, but I also spend a lot of time at the S2C2 Center and interact with the wonderful people there. It's a privilege. And what I'll be talking about today is I'll start with just a very quick sort of background on cryo-EM ET, so we're kind of on the same page. And then I'll get into building and fitting molecular models into these maps, which is something that I've been working on for many years. And I'll get into map model scores, which help us to assess how well maps are resolved, how well models are supported by the experimental cryo-EM maps that we see. And in particular, I'll talk about the Q-score, which is something we've been developing at Stanford and is gaining some traction, applying it to proteins, RNA, ligands, all types of small molecules. And it's starting to be used in the EMDB validation report when depositing a map and model. So I'll go over how it's being used and what the future prospects are looking like. So just a very quick sort of overview of cryo-EM as we all know it. So we have a frozen sample of particles. So we pick the particles, we do 2D classification, and then finally try to find the poses of each image to build a 3D model using back projection. And with this 3D model is often refined at length by going back to the particles with various processes. With CryoAT, on the other hand, which is starting to produce maps at higher resolutions as well, using sub-tomogram averaging, we have cultured cells or on a grid. The grid is frozen. A lot of times a lamella is formed to create a thinner sort of plane that can be imaged better. And this lamella is actually tilted and allowing a 3D volume to be built using reconstruction methods. And usually with tomography, the final step is to do annotation or segmentation to try to identify different features in a cell. Recently, sub-tomogram averaging has become more popular. So here, when you have a tomogram that's annotated, you can see various features, membranes, different cellular structures. Sometimes these structures can be repeating. So for example, we see these filaments that presumably have repeating units. And if they are repeating, we can extract these as particles and then create sub-tomogram averages, which go to higher resolutions. So for example, this particular fibril was averaged to about 9.3 angstrom resolution. And this other linear sort of fibril that turns out to be a microtubule was averaged to a little bit higher resolution of 6.7 angstroms. And in these maps, there is some limitation as to what we can see, but a lot of times we can fit known structures, identifying secondary structures and their positions in the complex. So these are starting to become more useful in identifying cellular structures in actual cells rather than purified. Then I want to quickly talk about resolution because this is something that's related to map quality and the Q-score as well. So the way that we measure resolution in sub-tomogram averaging and recently and more traditionally in single particle reconstruction is we have these particle images and they're split up into two half sets. They generate two different half maps. And we can see sort of the detail in these half maps by turning them into Fourier space. And then we look at increasing frequencies starting from the center and radial shells. And we compare the values in these Fourier transforms via this Fourier shell correlation plot, which tells us how well these two maps correlate at increasing radial spatial frequency distances from the center. And we see that these maps in particular agree very well at smaller spatial frequencies, which means that which tells us how well the signal correlates. Eventually, as you go to higher spatial frequencies, they start to correlate less and less, which means that the noise overcomes the signal. And here, this tells us roughly what resolution the map is. And this is similar to, but not quite the same as what is typically meant by microscopy resolution, where, for example, a resolution of 2 angstroms tells us how far apart two objects can be and then still be able to tell them apart. So that's the main idea of this, of doing this FSC. And the trouble with cryo-EM being is that the noise eventually starts to dominate. So, and also just to quickly overview different structures that we're looking for in these maps. So for proteins, we have the primary level, which is just the sequence. We have secondary structures, alpha helices, beta strands. The tertiary, the tertiary structure level tells us the overall shape of the protein, and quaternary tells us how different proteins assemble together. We also started to look more at nucleic acids or RNA. So in terms of RNA, at the primary level, we also have the sequence. The secondary structure level is more of which nucleotides interact via base pairs with other nucleotides. So you'll often see these diagrams, and they have slightly more complex shapes. So and different terms are used for these. We have helices as well, loops, various types of loops, and so on. At the tertiary level, we also have just the overall shape. And then we can also have assemblies, including multiple RNA fragments or RNA proteins. So this is what we're looking for in terms of how to interpret these maps from single particle and sub-tomogram averaging. And what we can see at different resolutions. So for example, this map at 13 angstrom resolution, Here we see more the quaternary level, so we can, if we're careful, we can potentially fit different proteins and see how they fit together. At a slightly higher resolution, surpassing 10 angstroms, we start to see the secondary structures, helices and beta sheets. At 3 angstrom resolution, we should be able to see individual strands separated in the map. We start to see the helix pitch, so you can see a little curve, little indentations where the helix is curving. And then we can also see loops and side chains. So especially the bulkier side chains, we might not see some side chains that are more flexible, or it could be radiation damage like this aspartate over here. And getting closer to 2 angstrom resolution, often you will see much more clear side chains. So you see the contours of side chains. We might start to see even some densities that could be attributed to ions. At higher than 2 angstrom resolutions, we can start to see donut-like shapes for the aromatics. We see more clear ion and water. And even you'll notice that the contours of these atoms start to become a bit more clear. So it becomes a lot more easy to build models and be confident in their configuration. With RNA, at something like 10, 11 angstrom resolution, we kind of see the overall shape. It might not be easy to identify the different base pairs, but a lot of times a model can be fitted as well. Closer to 5 angstrom resolutions, we can start to see that the ribbons have much more clear shapes with these indentations in the ribbon, these helical ribbon-like structures. Closer to 3 angstrom resolutions, now the actual nucleotides become a lot more clear. So you can sort of see that each nucleotide is separated, and then you can sort of identify which ones are, or the base pairing between such different nucleotides. Much like with proteins, you can start to see ion densities, and there's lots of ions and waters around RNA. It's something like 2 angstrom resolutions. You can see waters even more clearly. So you can see that waters play some role in even base pairing like interactions. You see here that there's this base pair is actually augmented by a water interaction. This is 2.2 angstroms is so far, I think, the highest resolution achieved for a single RNA only structure. But there are maps with higher resolutions, close to 1.3 of the ribosome. And here, much like with proteins, we see holes for aromatics and nucleotides and much more clear water and ion densities. Okay, so now let's get it how we actually model these maps. So if you have a map that's relatively high resolution, the go-to for sort of interactive building is still Qt. I think a lot of people, and we also use Qt a lot. And in Qt, if you see the sort of protein backbone, you can start to build a model using these little widgets. So here I added a residue. It won't know by itself what the actual residue is. So here, this looked a bit like a proline. So this was mutated to a proline. But you'll notice that it's actually not too hard to kind of trace the backbone and build a model that way. Some side chains like this one might be a little ambiguous. This one looks like something bulky, like an aromatic. So likely a phenylalanine. So you can mutate this to a phenylalanine. So here, knowing the sequence is actually very critical, really, for building models like this. And then you can do refinement to kind of go from the initial guessed configuration to something that fits a density reasonably well. More recently, there's been more automated tools to do this. So for example, one recent method is being Model Angelo. So this one will basically automatically try to identify residue positions in the map. And then based on these residue positions, it will try to join them up together using a graph-like approach with nearest neighbors. This graph is then optimized with various tools, neural networks. And if you provide a sequence, they can actually also try to guess where the exact sort of position of each residue from that sequence in the map. So it actually does quite well for maps that are high resolutions, 2 to 3 angstroms. Lower than 3 angstroms, it can start to be a little bit more sort of incomplete. You get a more incomplete model. So a lot of times you might have to go back to Qt and tools like that to interactively modify it. Now, for low resolution maps, so let's say, or even for high resolution maps, a lot of times it helps to have a starting model. So if you have your map here and you have some known model that you think is in that map, we can try to fit this into the map first. And we do this usually either interactively or using exhaustive search tools. So interactively in ChimeraX, you can just sort of manually move this model to where you think it fits and then do a quick sort of rigid refinement. With exhaustive search tools like Citus, Phoenix, Doc and Map, and even ChimeraX, you can try to look for sort of exhaustively go through all the positions and orientations of the model in the map and see which one has the best score, and then use that and then look more carefully to see if that's really the correct position, which is sometimes a bit of a challenge to know if the top scoring fit is actually the correct one, especially as you go to lower resolutions. At high resolutions, if the secondary structures match well, then you can be pretty confident with these that you're finding the correct position. Sometimes when doing rigid fitting, you might notice that the model is actually not follow the map densities quite well. So in that case, we can use various refinement tools. This is a molecular dynamics-based tool, MDFF, which has been around for a while and more recently is old, is actually quite nice for interactively doing this in ChimeraX directly. But you'll see that if you start if the model is close enough to the densities, it can sort of be pushed into the density so that it matches the density better. And with Isolde, what's nice is it's interactive, so you might have to pull and push and pull some of these atoms to try to drive them towards the right configuration. And okay, so now when evaluating a model or when doing refinement, there's actually some model parameters that we use. And these model parameters include bond lengths, so how far apart two atoms are that are covalently bonded, and bond angles. And these parameters are often, or the more standard parameters used are actually derived from known structures, known high-resolution crystal structures. So from these structures, we can sort of characterize what is the equilibrium or what is the most commonly seen bond length or angle between any two or three types of atoms. And then we try to drive them towards that equilibrium value. And then we also have some statistics, so standard deviation in the observed values. So at which point do they become less and less observed, which means that they're likely less energetically favorable. So when we evaluate whether a model has good structural properties, if some of these bond lengths and angles are really far away from that equilibrium value, we usually say that that's less reliable or less or lower, less confidence in the model. Another parameter is a dihedral. So a dihedral is a rotation. You can think of it as a rotation around a bond. It involves, to define a dihedral, we usually use four bonded atoms. So we start here, then this one, and it's basically the angle between these four atoms. We also use van der Waals radii. So if two atoms are too close together, that means that they're very likely to have this clash. So they don't like to be that close together. So clash scores are also measured to see if any two atoms are too close together to be physically realistic. And another important property are these special backbone dihedrals, which include the phi and psi angles. And these phi and psi angles are actually plotted often in these Ramachandran plots. And again, based on a lot of high resolution structures, we see that these phi and psi angles like to be in these particular areas where they're commonly observed. And any sort of phi and psi angles that fall outside these regions tend to be characterized as unrealistic or sort of not good for the model. This omega angle is actually a special angle, and this one usually has to be either 180 degrees or for a normal CN bond, or 0 degrees, which indicates a cis peptide bond. It was what's known as a cis peptide bond. So 0 and 80 are seen. 0 is seen less commonly. So cis peptide bonds are usually kind of looked at with caution. But any sort of omega angles that are far from those values are also generally not thought to be good. We also characterize different sidechain configurations via rotamer libraries. So there's a very widely used Dunbrack rotamer library, which tells us that for a given set of backbone Phi and C angles, what are the likely conformations for that sidechain. So we use this a lot also when model building and also when evaluating models. So if the configuration that is in the model is kind of not close to any of these sort of widely observed configurations, and it's also thought to be not great. And finally, one thing that we're starting to see more recently with higher resolution structures is that oftentimes you can have just one single conformation for a side chain, and here we can see it's well resolved in the density. But there's also cases when it's less resolved that you see sort of evidence that there's actually more than one possible rotamer or alternate conformation for a side chain. And okay, so a lot of times we'll use this report to kind of get like an overall picture of how good the model is based on all these parameters I talked about. So we look at the clash score. So if the clash score is low, that's a good thing. It evaluates whether there's a lot of good or bad rotamers. So generally that's thought to be good if there's more favored and not too many poor ones. Ramachandran outliers kind of tells us how good the backbone configuration is. There's also the bad bonds and bad angles, which here you want to have these two as low as possible. So 0, 1 is ideal. The goal is 0% for bad bonds because it's thought that atoms really like to be a certain distance away and not much further than that. There's the cis-prolines and twisted peptides referred to the omega angle. So if they're far from 180 or 0, that's considered too bad. And actually, this is how important that parameter is. Even one twisted peptide is actually flagged as red. The goal is 0. There is some flexibility around that 180 and 0 angles, as I was showing before, using the standard deviations of the observed values. So usually it's anything within 4 or 5 standard deviations is thought to be okay. And there's more parameters here that involve different methods of evaluating the model quality. Okay, so another thing you'll often see when depositing a model is the EMDB validation report. So here, much like with the mole probability report, which is they're all based on the same methods of evaluating the model quality. It will report the clash score, Ramachandran, and sidechain outliers. So sidechains are the rotamers that we saw, and again, it sort of evaluates how well does this model. How good is this model compared to the scores obtained for other models at similar resolutions? So it's percentile relative to all structures or percentiles relatives to EM structures only. And again, sometimes this helps to indicate whether what you're depositing is good, or maybe if it's in the red, you might want to do some more refinement or look at different parts of it and see if the scores can be improved, which the idea being that the better the scores, the more confidence you can have in your model. So far in this overall validation report, there isn't yet a map model score. But as I was mentioning, the Q score is starting to be used later on in the validation report. So we'll look at how it's starting to be used. First of all, a quick intro on map model scores. So actually in the EMDB validation report, you will see an atom inclusion score. And with atom inclusion, it basically just looks at whether each atom is inside the density or inside this contour at a given map contour level. So it uses what is the recommended contour level for the map given by the depositor. And if you have many atoms that are inside that contour, you get a good score. If there are many that are outside, you get a lower score. With sort of the limitation being that it does depend highly on that contour level. So you could specify a very low contour level and include more atoms. So it's been taken with a bit of a sort of cautiously in the past. We can also, another very basic score is the average density, and this is used a lot in refinement, but the average density just tries to maximize the the map value at each atom position without really looking at what's going on around the atom. Slightly more sophisticated is the cross correlation score. So here we know here we first generate a map of the model. So the gray surface is the CryoEM map, and the blue surface here is a map generated from the model. And usually this is done by placing a Gaussian or small number of Gaussians at each atom position. And so you'll see that these atoms are actually, or this map looks Gaussian-like, and the width of the Gaussian is relative to the resolution. So if I want to simulate a map at low resolution, I just use a wider width for the Gaussian. And there's various formulas for converting the width of the Gaussian to the resolution. So that's not, but it's still kind of approximate. So that's kind of one parameter that's sort of it's involved in this process. The other consideration is that there's actually two different formulations that are used for cross correlation. So there's one where it's just a simple dot product between the map values in the simulated map and the map values in the cryo-EM map. So this is just a simple sort of correlation normalized. So you get a value between -1 and 1. And there's also this other formulation, which is known as cross correlation about the mean. And here, if we think of all the values as a vector m, we subtract the mean of the vector from that vector, and then do a dot product between those two. And these two formulations can give slightly different scores. So it's kind of, and each one has advantages and disadvantages that we've looked at in the past. I won't get too much into that here, but it's just good to know that there are different these two different formulations that are commonly used. When we got to higher resolution maps not too long ago, we started to look a little bit more closely at what's going on around each atom in a cryo-EM map. So for example, in this map at 1 angstrom resolution, the atoms are well separated, so you can resolve individual atoms. So we looked at the map values at increasing radial distances away from this atom. And we noted that these map profiles are Gaussian-like, which shouldn't be too much of a surprise. That's how we thought each atom would look like. And we can even fit an actual Gaussian with this dotted line as a Gaussian function. So we can fit a Gaussian function to this, and it turns out to match pretty well. It has the tail kind of trails off a little bit, but for the most part, it was somewhat surprising how well even a single Gaussian can fit this curve. So a Gaussian has this formulation, it's an exponential, and the parameters are this sigma, which gives you the width of the Gaussian. It's often referred to as the width. And we have these height A and B parameters. So A and B, what we see with low resolutions, sort of taking out the A and B and kind of normalizing them, we see that indeed, even as you go to lower resolutions, using this sort of map profile sort of method, you see that even at low resolutions, they're still Gaussian-like, so we can fit different Gaussians, and each Gaussian has a wider width. So just like I was mentioning before, if you want a map with low resolution, generally you just use a wider Gaussian. So that's what we see here as well in these experimental maps going all the way to 2.3 angstrom resolution. So we thought about how to turn this into a score. So we thought that characterizing, okay, how Gaussian-like is the shape, and also how close is it to sort of this idealized Gaussian that we see in a high resolution map. So that we kind of consider these two things to calculate the Q score. That's how it came about. So we compare the map profile calculated for each atom in the map to this reference Gaussian, which we see roughly at 1 angstrom resolution. So if we compare them for this map and model, we see that we get a Q score of close to 1. And then as we go to low resolutions, 1.3, now the map profiles starts to get a little bit wider, slightly lower Q score, and even wider, again, lower Q score, and 2.3, even further away, so lower Q score. So this started to work somewhat, and the Q score is calculated much like cross correlation. It's actually using this cross correlation about the mean formulation, but where we take map values and values U in the map and compare them to the values we would see with the actual perfect Gaussian. So that's how it's calculated via correlation of the map values with the reference Gaussian. So it allows us to get scores like this. So if an atom is well resolved, again, we see a high Q score. And if the atom, for some reason, whatever it may be, is not resolved, the density is weak or noisy around it, usually you'll get a lower Q score. And the map profile for this atom, which is outside the density, looks like this. And this one, as before, it has this green curve, which is Gaussian-like. So far, for an example like this, it seems to work good. And more recently, we applied the Q-score to a lot of maps and models in the EMDB just to see a little bit better how it fares at different resolutions. So here we take the Q-score, we calculate the Q-score of every atom in every model, and then for each model, it's averaged and it's plotted versus the reported resolution of the map. And much like we sort of saw before in earlier work, seems to be a very good correlation between the Q-score and the reported resolution, meaning that the Q-score is actually good at sort of capturing just the resolvability of different features in the map, much like what the reported resolution tries to do, and also visually. So as you go to lower resolutions and lower sort of visual detail, the Q-score also gets lower. So that's a good thing. And it can even be applied in this, it's not as in this resolution range between 5 and 10, you'll notice that it decreases very slowly, so it's not quite as useful. But still, the lower sort of the visibility of different features like these alpha helices, the Q-score still kind of decreases slightly in that resolution range. Now, there's a lot of points outside. So we started to think, okay, is there a way to kind of statistically identify points that are maybe not, there's something strange going on with? So we calculated these 95 percentile lines, which we call a Q high, so the high 95 percentile and then low 95 percentile. And Q peak we refer to as sort of the peak of the distribution. So that tells us for each resolution, what is sort of the most commonly observed Q-score value, given all these 10K maps and models that we've looked at so far. And there's a lot of these points here outside these lines, which are, we kind of want to see what's going on. So for example, this example one, it's a map that's reported to have about 2.3 angstrom resolution, but has a very low Q-score. So when we look at the map and model, we see that there's a lot of detail in the map, so that's not the problem. The problem was it just wasn't fitted. The model wasn't fitted very well. It was just slightly shifted. So the fit to map also affects the Q-score. So that's not fully separable really. So the Q-score itself won't tell you the quality of the map, but it will tell you a combination of how well the model is fitted. If it's properly fitted, it can tell you, as we saw before, sort of the quality of the map potentially. But here with a small sort of quick rigid refinement, we get a more expected Q-score level of 0.7. So that's good. This point has a very high Q-score, much higher than what is commonly observed for this resolution range. This also seems strange, but there's not quite as many dots that are above rather than below. So it's harder to get higher Q-scores for a given resolution than it is to get lower Q-scores. But with this point here in particular, it was kind of interesting because if you look at the map, it has this broken, noisy-like shape or sort of features. And this tends to indicate over-sharpening, which is possible. A lot of times with sharpening, we try to boost the detail in the map by amplifying the higher frequencies. But if you amplify too much, you're amplifying noise, and you see this noisy, broken up contour as here. And we've also shown that Q-scores can actually be influenced by this. So if you over-sharpen, you can get slightly higher Q-scores. And it's even worse if you over-sharpen and then you refine, you're driving atoms towards those noise peaks. So you can get slightly higher Q-scores in this case. So this is an interesting example because you can identify, potential over-sharpening and over-refinement for a given map and model, which is not really great, really. For something that's low resolution, so for example, this 0.3, it has a low reported resolution, but it has a Q-score of nearly 0. So even though, as I was saying, the Q-score, is maybe a little bit less useful in this resolution range, if you get a Q-score of 0, there's still kind of something suspicious going on. So if we look at this, again, it seems to have the model didn't really seem to be fitted correctly. This one, to fit correctly, involved a shift and a rotation, but with the proper fit, you get a more expected Q-score level of 0.23. And then finally, this 0.4, which was also kind of above the line, seems to have a very high Q-score for this resolution. So here we can see that in this map, there are parts that are well defined and should have high Q-scores, but then there's parts that are much more smooth and lower resolution. So it seems like the resolution estimate here was maybe biased towards these parts. So again, kind of looking at points that are outside, this can sort of flag sort of these inconsistencies. So this map can be seen to have more resolution than what the reported value seems to indicate. Okay, we can also use Q-scores at a residue level. So for example, for this map and model, we can calculate Q-scores for each residue. And then we can plot these Q-scores at a residue level on the ribbon itself. So red means lower Q-score, blue is higher Q-score. So this can sort of visually help you identify parts in the model that might not be either not resolved or not fitted properly. So that can help to find problems and sort of guide refinement. In the plot here, we plot the peak sort of line. So Q-scores that are close to this line, this is probably as good as we can do in terms of fitting the model to the map. And then points that are far outside these lines, can indicate problem areas, as I was mentioning. So that's how we can use these plots. And in particular, in this area, we can see that actually the map seems to be resolved okay, but maybe the model is just not fitted optimally yet. So we use this a lot before depositing a model to try to find better models that fit better. We can also apply it to RNA. So in this case, it's calculated at a per nucleotide level. So when we have a low Q-score, or for here red, we can see the map is not that well resolved. There's probably not too much we can do in terms of fitting the model better in this case. And then there's some other parts, again, that are slightly red here, which might be a little bit more dynamic and less resolved. So these per nucleotide Q-score plots can also help to identify those areas. So this is indicating a region where it's resolved as expected, which is shown here. And this is a region here in A, which is less resolved. So it can help to sort of guide interpretation. So if you're trying to describe how this RNA functions, there might be less information to base on in these parts with lower Q-scores. Can also be applied for small molecules. So for example, these glycans. So we can plot per glycan Q-scores, and then this can indicate which glycans are resolved well. So these all have very good Q-scores. And then there's some that are not resolved too well. We can build these glycans based on mass spec data, just to kind of see what they look like and if there's potential interactions. But again, the Q-score can help to guide which ones we can sort of interpret with confidence. And then finally, I'll show this Q-score sort of example for a small ligand. So recently we had a ligand challenge, which was part of a validation sort of effort between different groups. Really, we try to involve the entire community, but we basically proposed this map amongst three maps in total, but this was one of the targets. And then we sort of asked participants to build models for the ligands. So this was actually the deposited ligand. And we can see that it fits the density reasonably well. The Q-score was actually a little bit low when we looked at it. So this Q-score of 0.6 is actually kind of on the low 95 percentile range. So this kind of model, I guess, could have raised a flag in terms of the Q-score, but again, initially sort of looked reasonable. In the challenge, we saw that participants proposed actually two different conformations for this ligand. So one conformation had this O5 atom, you'll notice here, kind of in this pointing down. And then in this conformation, actually that whole aromatic is flipped to around 180 degrees. And that O5 is actually pointing up in this direction. And so it's a little bit puzzling at first as we're evaluating the results, which one was the right conformation? Could it be a combination of two different conformations? This one seemed to fit the density overall better. This one seemed to have like a small space here that wasn't explained by the model. So, but in terms of Q-score, interestingly, the Q-score was a bit higher for this model, and it doesn't the display. It's hard to see in the display here, but actually these bumps, these small atom contours, actually indicate that this conformation is actually like the atoms fit better in the contour in this case. You'll notice here there's another space, there is another contour here for this atom. There's a nice contour bump for this atom and so on. So in terms of Q-score, this seemed to be the more likely conformation. And then we also noticed that there's actually a map that has a similar reported resolution, but seem to have slightly better visual quality. And this can happen. You can have maps that are reported to have the same resolution, but one might look better than the other. In terms of Q-score, it was actually, if you look at the Q-scores of the residues around the ligand, they actually are a little bit higher in this map, sort of agreeing with that visual sort of observation that the map is slightly better quality. The Q-scores were also higher of these residues. For this conformation, the Q-score was even lower, and then it sort of seemed to indicate that this conformation could potentially be problematic. And when we look at this conformation here, we get a Q-score that's in line with what is the expected value. So I guess the final takeaway from this was that it can be really useful if you're sort of modeling small ligands when it can be hard to know for sure whether the conformation is modeled properly. It can really help to look at Q-scores as sort of quantitative measure to assess how confident you can be in that conformation. And then finally, so I've talked about the EMDB, the validation report. So you'll notice further down in the report, as I mentioned, Q-scores are starting to be reported. So you'll see that it's plotting this per residue color-coded model, and with red meaning lower Q-scores and then blue being higher Q-scores. And it's just the starting point right now. Right now, there's not yet a guide as to what is a good Q-score. And as I talked in this presentation, we think that what a good sort of guideline might be is, okay, well, what is the most commonly observed Q-score for a given resolution? And if you're getting close to that Q-score, that means that you can be confident you've done sort of as well as you can in fitting the model. And then it's also sort of seems to say that the reported resolution is in line with the quality level of other maps at similar resolutions in the EMDB deposited so far. So this is something that's being worked on right now. We're collaborating with the EMDB team to kind of create better, to help interpret these scores overall. And they're also reported in the validation report at a per chain level. So much like as I was showing before with a plot, but this is the overall Q-score for each chain. So each chain can represent either a different protein or different RNA segment. It could also represent different small molecules. So you can look at what the Q-score for those are, and it's also reporting the atom inclusion. But yeah, the Q-score can be useful to, again, comparing it to sort of what the expected value is, can help you to determine if you should look at each chain perhaps a bit more carefully and so on. And and so that's pretty much it. In terms of outside the validation report, we have these tools in Chimera and now in ChimeraX. So in Chimera, it can help you to visualize these per residue Q-scores. So you calculate the Q-scores, then there's a button that will basically color code the ribbon, so you can use that as a visual guide. And with ChimeraX, even more recently, you can actually directly use Q-scores in combination with Isolde to kind of interactively refine the model in areas where the Q-score might be too low. And it can be applied to small molecules, as I mentioned before. But yeah, the final takeaway is that the Q-score can be a useful tool before depositing or as you're building a model to kind of tell you, is the model fitted properly? Could it be potentially better? And just looking at it to make sure if the map is not resolved, there's not much you can do, but then we can use the Q-score as a confidence level. So if the Q-score is low, it might be better to more cautiously sort of use that in sort of the interpretation of the map and model. So that's it. Hopefully that was interesting, and I'm open for some questions if there are any.
So we have one question so far.
Mhm.
To make sure everyone sees this as I was going to ask. So how do you obtain a map with the Q-square values being used to map the colors on the map? How do you switch the color score, the color scheme being used to be based on the Q-square value?
Ah, okay. The color scheme. Yeah. So that's a very good question. Actually, I think I made at one point a tutorial for doing this in Chimera, which is on YouTube. It's actually not super easy. You can change the color key. Um, right now it actually might be restricted to red, blue. But yeah, that could potentially further be adjusted to allow for using different colors. I think right now it might be restricted to red and blue in the tools. Yeah. So if you're interested in that, you can just maybe send me an email and then we can look into it. It wouldn't be too hard to add or adjust.
Okay. Let's see here. For a large molecule with low and high resolution regions, uh, focus classification, focus classification and refinement gives good maps for model building. Would it be a good practice to use a composite map of the focused refinements to calculate Q scores? How do you handle this?
Yeah, that's a great topic. I think that's being discussed a lot recently. So the Q-score doesn't care too much whether it's a composite map or not. It really will kind of evaluate just how well atoms and sort of groups of atoms are resolved. So I wouldn't sort of I wouldn't think that it's a bad idea to calculate it. I think it's a good idea just to kind of it can also tell you again, because it's really comparing it to what an idealized atom should look like. So the closer to that idealized atom that map is, the we think the better the quality of the map. That's the main sort of theorem. So this can also, yeah. So there's a lot of questions as to whether these maps, kind of capture these atomic details, or somehow the averaging process might sort of disturb that actual quality of the map. So this might be a good indication as to whether these methods are actually working well, or maybe need some adjustment in how it actually composites the two maps together. Um, and that sort of gets into this other, yeah, into this other sort of um, yeah, no, I think so. Yeah, so I think it's a good idea to do it. Yeah, and then to see, because ideally the higher the Q score, as we've observed, the better the resolvability of different atom groups. So I think it can be very useful in that case.
Hmm. Okay. Um, Another question here is, does PDB consider showing Q score on structure summary pages together with class score, Ramachandran, outliers, etc.?
Yeah, so in terms of the overview, that's something we've talked about recently, and I think it's in the works because it would help to have sort of an overall map quality or map model fit sort of quality along with those other model parameters. Right now, just that overview is only showing, yeah, those three parameter sort of categories that are just only model oriented, right? So clash score is only model oriented. So, so yeah, ultimately, I think the goal will be to have on the summary page at least also this indication of map model fit, and I think we're kind of seeing how if we can use Q-score for that. And I think with the recent work on showing these statistics for a lot of maps models in the EMDB, I think that's helping to guide that development. But hopefully, yeah, and I think that would be a very good thing to show. Um, yeah, so I think we will see a bit more of that in the near future. But for
Okay.
now, there's no, right now you can do it, using these tools, using the Q-score tool. So you can do it sort of separately. Again, the idea being that, like when you deposit a map and model, it's really the EMDB is not going to stop you from depositing a map and model. that you want to deposit. But the idea is that if you want to try to make the map and model as good and as have a high confidence level as you can, then using all these different tools is good on your own, right? That's kind of how it's the main idea. It's not really a way to kind of prevent people from depositing maps and models. The key is still to deposit them, and then as we go on, we'll have better tools for assessing all these properties.
So related to deposition, is there a recommended method to identify an appropriate map contour level for atom inclusion calculations?
Yeah, that's an interesting question. I think it's done mostly visually at this point. Yeah, I guess I don't think there's any method that I know of that you would run and it would tell you here's the perfect contour level. Um, but yeah, it's an interesting question to think about. I don't think, to my knowledge, I don't think there is a tool like that out there. But you might try something like, optimizing sort of noise, how much noise you see versus how many atoms are inside the contour level. So you might try to decrease the contour level to include as many atoms as possible while not adding too much noise, right? You'll notice as you lower the contour more and more, all of these background noise start to appear. So something like that could be potentially done. And I think visually that's what we do is we kind of decrease it to as low as we can. And then when we start to see too much noise appear, then we stop. I think that's kind of the sort of ad hoc method, but I don't know of any methods to do it automatically.
Okay. And then for a flexible region, which usually gives us relatively poor quality map, what would you recommend for balancing for understanding that between the Ramachandran plot and the Q-score? Which do you think is better?
Well, I guess the Q score won't tell you much. If you get low Q scores in these regions, like these ones here, it probably won't be too useful. So then in that case, the model properties do end up being the model parameters will be more useful to tell you if it's a physically realistic model or not. Um, the thing with models is even in a small region like this, the number of confirmations can be really high, as any modeler can tell you. So the choice of these sort of loops and things like that that are dynamic, it's pretty tough to model. I think a combination of Ramachandran is good, but one bad Ramachandran angle or residue doesn't mean your model is wrong. It could be like I guess so. Yeah, so it's hard to say exactly. The Kablam score that we saw in the mole probability can also be useful, but I think that's more useful for areas that are like have secondary structures. Um, Looking at interactions can be useful. Is there hydrogen bonding in those areas? So I think, yeah, the model parameters will be more important in those regions. Yeah.
Greg, if you don't mind, I'm gonna pop in with a question. I can't type in the Q&A.
Mhm.
So you're using the Q score as a validation metric. And what's really awesome about this particular validation metric is none of the refinement programs are using it. So it is like truly a validation metric, which is absolutely gorgeous.
Uh-huh.
But I could imagine you could use this idea to come up with a new resolution metric. And is that something you've thought about? Like could
Yeah.
you define resolution by Gaussianness of a map?
Yeah, you could, and that's actually the first thing I tried when I was fitting Gaussians to the atom profiles. I thought, oh, okay, I can just fit a Gaussian, and then I'll know what the resolution is for that atom. It worked okay in a lot of cases where the atoms are well defined, but if the atom is not well defined, then fitting a Gaussian didn't work so well. It wasn't very robust. Um, so yeah, so that's definitely sort of seems to be, yeah, but and then the other caveat being that if you have a very sharp peak, you might think, the map is so well resolved, but that could also be potentially noise. So there's some of these sort of pitfalls in calculating Q scores. And I think in terms of using that as a resolution estimate, I think these kind of things can sort of detour it, or what's the word? Kind of confuse it, right? Because it might think, oh, if you have an atom fitted to a sharp noise peak, you might say, oh, the resolution is really high. So, but on average, yeah, on average, I think they're very interchangeable in a way, right? As we saw from the sort of the regression line, you could potentially use them sort of interchangeably, ideally. And we saw that in maps that were reported to have the same resolution. One had slightly better detail. The Q-scores were higher, so that can tell you, yeah, this map is actually better than the average at this reported resolution. Um, and this seems to work better and better with the more atoms you include, right? So the more you average, then this seems to be more reliable. If you're averaging over a small number of atoms, like a backbone group, like or a side chain, then it might be hard to relate this directly to resolution, again, because each area might be a little different. There might be more flexibility. So that can be less of an indication of the resolution of the map. But yeah, taken together on average, seems to, yeah, because they're correlating so well, they can sort of, yeah. And again, with the other assumption being that the model is fitted well. So there's all these combination of things that go into it, which I think it makes it hard to say that for sure this Q score has this resolution. There's all these other things going on that we have to be careful with.
Great. And it looks like we've gotten to all the questions and we're right on the hour, so we can finish up on time today. So thank you for that awesome talk. We haven't talked a lot about validation in our webinar, so that was a really nice area in the workflow to go into. Thank you so much. And
Great.
thanks to everyone for joining us today. The national centers are here to support your science. We wish you all well in your cryo endeavors, and let us know how we can help. And we will see you all here next month.